Hey, good afternoon, everybody. Welcome to our today's inaugural lecture. It's my great pleasure to welcome you all to what is the very last one in this year's series of faculty inaugural lectures. And I think most of you, but probably not all of you, know who I am. I'm Professor Steve Darby. I'm the Associate Dean for Research for our faculty. So I work together with the team in FOS on coordinating these sessions. Just at the outset, I wanted to welcome you all. Thanks for coming, whether you're in the room or online, and just say very quickly that these are a real pleasure to host, and I'm delighted that there's a good turnout in the room today as well for your talk, Owen. I mean, I think these are really important events because they're an opportunity to celebrate success. They're an opportunity to hear about fabulous research that colleagues do in the faculty. And the final thing that we like to do with these lectures is that we do record them, so be a little bit careful what you say, and we put them up onto the website, the inaugural lecture website afterwards, because inaugurals are a little bit of an odd thing in a way. They're unusual lectures in that they kind of talk about cutting edge work, but in a way that's accessible to people, including perhaps people not only in other disciplines, but maybe the public more broadly. So one of the things that we're hoping will happen is that as we have more and more of these lectures, we're building up over time quite a nice resource on the website that showcases what we do across the faculty to the general public. So they're the reasons that we do these things. I'm going to stop there. I'm going to hand over to Phil Williamson, the head of School for Biological Sciences, and to introduce our speaker today. So over to you, Phil.
Okay, so it's a great privilege to introduce Owen Rackham, Professor Owen Rackham, this afternoon. So I thought I'd provide a little bit of a background for those of you who sort of are less familiar with his work. So Owen undertook his undergraduate degree in computing science before starting a transition to biological sciences with an MSc nature-inspired computing. So he subsequently embarked on his PhD in complexity science at the University of Bristol from 2009 to 2012. And in 2012, he made the leap to Imperial College London, where he joined as an MRC Career Development Fellow before moving to Duke NUS Medical School in Singapore as an assistant professor. We were fortunate enough in 2020 to recruit Owen to the School of Biological Sciences as an associate professor. So on that long journey, his research group has been undertaking some pioneering work, understanding what defines a human cell, unraveling how their properties from genome, transcriptome, and the surrounding environments determine the cell's fate. So his research is really is highly interdisciplinary, so it really spans many different disciplines, bringing together experimental approaches with computational approaches, using the latest developments in artificial intelligence to understand the complex interactions within the cells, which could lead to their final fate. So, Owen's had a long career in academia, but at the same time, he's also had a presence in the sort of the enterprise space. So, based on some of Owen's early work, he was successful in co-founding a company called Mogrify that deploys next generation sequencing, gene regulatory, and epigenetic data network analysis to predict the transcription factors and growth factors that are necessarily to determine a cell's fate. And that company, I think, can go from strength to strength from what I gather. So since returning to the UK, Owen's played a key role in the life sciences landscape in the UK. So from 2022 to 2024, we lost Owen again briefly to the Turing Institute, where he was the theme lead for the cell and molecular sciences theme. And since returning to Southampton, he's continued in that similar vein under the auspices of the Institute for Life Sciences. And we're very happy that Owen shares a lot of this insight with our undergraduates, contributing to the sort of the computational biology offering within the school. And I know Owen and his group are very, they're always keen to sort of get these types of artificial intelligence methods used more broadly within the school, organising lots of training workshops, and for that, I think we're all grateful. So Owen was promoted to professor in 2025, so you've had to wait a while for your inaugural lecture. But since 2025, at the start of this year, we lost Owen, or we lost half of Owen again, as he set up a really fascinating collaboration with the Rosalind Franklin Institute, where he's on secondment. And I think this is a fantastic opportunity. It's one of the strangest meetings I've been to, is going to somebody else's job interview, but it was an interesting experience. I think it's a fantastic opportunity for Owen to link up his expertise in computational biology, artificial intelligence, and link sort of the omics technologies to the spatial technologies that are being developed at the Rosalind Franklin Institute. So without further ado, I'm going to hand over to Owen so he can tell us more about this fascinating research that he's doing. So over to you, Owen.
Thanks, Phil. Hi, everyone. Thanks for the introduction, Phil. Thanks for coming. I've stood at this lectern many times and given many lectures and never felt nervous before until today. So let's see how this goes, shall we? So it's really a great pleasure to be here. I've enjoyed a lot of my career, and it's nice that I get to show you some of it today. Perhaps not the sort of same way that you might have seen work that I've done before. What I will say is that picking what to talk about today was very hard, and picking how to present what I'm going to show you today was also very hard. But what I decided was to show you just one whole arc of a story, right? That started right at the beginning of like really when I went to university, right the way to things that are happening right now. That means that there are things that I'm not going to tell you about, and that's what I feel the most sorry about. But you're more than welcome to come and to ask me about those. So this is the title of my talk today, Learning the Rules of Life. But I thought we could start by going back in time. So here I am, looking pretty good. This is me. I'm doing what I liked to do the most, which was building stuff. And for those of you who were born after social media made things like this not possible, this is a bowl cut, which you won't see these days, but I look wonderful, as you can see. But at this point of time, nothing was more desirable to me than this. If anyone else had one of these, this is the Lego Rock Refuge set. It was a Lego set, and I love nothing better than building those things. And that's something that you'll see all the way through. I think that building things is something that I've really enjoyed all the way through. Okay, so that was me. Here's all of us looking great. And I was really very fortunate from the point of view of learning about science. My mum was a primary school teacher. My dad was a biochemist. So I didn't really have a lot of choice apart from to learn a lot about science. So I really enjoyed that, and it was a really great way to grow up and learn a lot of things. So safe to say that we were slightly on the geeky side. So my brother was a British chess champion, but if you think that's geeky, I was in a group which got named the British Harmonica Champion Group. So altogether, we were a pretty nerdy bunch. But one thing that came into our lives very early was computers, partly because my dad liked them so much. This is the first one that I can remember, the ZX81. I remember my dad typing in the code for games so that we could play them. And this was my favorite, Granny's Garden, in case anyone else also like those things. But that was really a feature, and I think that's probably why sometime in the 90s, I went here. I went to the University of Exeter, and I studied computer science. And here I am again, even worse than the bowl cup. But it was a really fantastic time to study computer science, actually. It was right about when the internet was becoming mainstream. We started to work out how to connect things together. Software engineering was becoming really sort of a discipline in its own right. Things like bioinformatics was starting to become a discipline in its own right. And I really enjoyed the work that I did. And I graduated. I graduated with a degree in computer science, had all of these things that I could go on to do, apply this great knowledge that I'd learned. So I took a job at a pre-packed salad company, which, as you can imagine, was the best motivation you could have for going back to university. So I left that job after a year, and I went to the University of Birmingham. And here I studied something called nature-inspired computing. So I went back to look at the things that I did. I'll show you. This is some of my first academic output. Fascinating title here. A multi-ant colony optimization algorithm for winter road salting problems. Try to stay calm. But actually, I went back and I looked at something else I did, which actually I'd forgotten I'd done it, but it was actually quite interesting. Not to say that the top one wasn't interesting, of course. I did a study. It was called Wavelets in Evolutionary Art, and actually, it was looking back at it now, looking at where we've ended up. It was a tool for making a representation of images, so you could make new images. So it learned from all these pictures of churches how to make a picture of a church. That's a church, I promise you. That's a face. But it was that was a really fun project, actually. And actually, these days, it's got a lot better, luckily, making images using AI. But it was fun to have a look back at that. Okay. But the thing that I really got out of that was this was a course in nature-inspired computing. And I shouldn't say this, but I had never done biology after GCSE up until this point. So I went here to the University of Birmingham, and we learned about things like evolution as a mode for doing computer programming. We learned about things like neural networks and the brain and how that inspired computing. And I realized that I'd really made a mistake by not studying biology. So sometime in the 2000s, I went here, the University of Bristol, and I did a PhD. I did a PhD in this centre called the Bristol Centre for Complexity Sciences. I still don't really know what that means, but it was one of the first DTPs that the UK ran, and I loved it. It was so much fun being in a cohort of students doing something all together. And it was also at that point that I met this guy. So this is Julian Goff, similar disregard for the barber as me. And he was my PhD supervisor. And he was really the kind of PhD supervisor that I still aspire to be, and I think everyone should aspire to be, because he just had such a passion for just doing things. And I found this photo, which I think demonstrates this the best. So, by the way, I was doing a PhD in protein evolution. But here we are on the roof of our building. This is shortly after they launched the International Space Station. And what Julian had found out is that if you build the right equipment, you could send a message to the International Space Station, and it would be stored on there, and it would broadcast it to the world until someone else replaced it with another message. So you can't see, but I'm standing here with an antennae, trying to trace the International Space Station across the sky, while Julian tries to send it a message. And like, this was the kind of thing that he just got us to do all of the time. So not only did we do what we were supposed to be doing, but he really encouraged us to play and think and try new things. And I've tried to carry that spirit through everything that I've done since then. So this was actually my first project. This is Spiracoil. It was looking at protein evolution. I'm not going to talk at all about protein evolution. I also did this other study at the beginning of my degree on the NMDA receptor. I'm not going to talk about the NMDA receptor at all today, because something happened during my PhD that completely changed the direction of everything that I was interested in scientifically, and everything that I wanted to do next. And it was because of these two guys. So I'm actually going to tell you about this guy first. I should use this so that everyone on the internet can see as well. So on the right, this is John Gurdon. He very sadly died at the end of last year, but he was really one of the most inspiring people that I met. He shared the Nobel Prize with this Shinya Yamanaka on this side. One of the things I love about John is he was always generous with his time, but also he very famously shared his school report. And in his school report, it said, I'll read it, I wrote it down here. It said, I believe Gurdon has ideas about becoming a scientist. On his present showing, this is quite ridiculous. Nobel Prize winner, John Gurdon. And I love, he had that sort of community. But the person that I really want to focus on is this guy over here. Here's a photo. I think he's awake. This guy over here, this is Shinya Yamanaka, and he made a discovery that really was the driving force behind a complete change of direction for me scientifically. But in order to explain why that discovery had such an impact on me, I'm going to try and give you a bit of an introduction to human development so that you can get a feeling for quite how profound what it was that he discovered. So I'm going to give you a 2-minute introduction to human development, bearing in mind that I'm a computer scientist and we're in a room with some biologists in it. But hopefully I don't do a complete misjustice to it. So I'm sure you all realize that we start as one cell, okay? But even at one cell, the complexity is quite mind-boggling, actually. So this is the Lego Star Wars Millennium Falcon. It's the most complicated Lego puzzle that you can buy. The catalogue number's there, in case anyone wants to buy it. This has 7,541 pieces. I've never built it, but I would love to. The catalogue number's there. That one cell just by itself has somewhere between 10 to the 9 or 10 to the 10 protein molecules. So this is the building that you came in. If you put all of those Lego pieces there, that's how many Lego pieces it would take to build that one cell, the one cell that we start with. But that's the beginning of the story. So that cell divides and it divides. And the first thing, we get this sort of compaction, this ball, which we call the morula. And then just after that, sort of the first, in my opinion, sort of interesting bit of biology starts to happen. So with some of the cells start to specialize. So basically the process of development is a process of specialization. The cells start off all the same. They divide and they divide and they specialize into things. And this is the first moment that we see some specialization. So this structure that I'm showing you here is called a blastocyst. The cells, they're in blue, they're embryonic stem cells. So they're the cells that go on to form the embryo. And the cells around the outside, they're the trophoblast cells, and they go on to form the placenta. I'm saying that because it's going to become useful later on in my talk, but that's sort of the first decision, if you like, that the cells make during differentiation. And then that continues. There's this process called gastrulation. The embryo forms, and then it starts to develop. And then after about 37 to 42 weeks, depending on how it goes, this happens. This is Daphne. She's over there. She's got a bit bigger. That's you, yeah. But by the time that Daphne was born, she was made up of trillions of cells. And as you can see, she's not just a bag of cells. Development is a very orchestrated process that means that the right cells end up in the right place, tissues form, structures form that are all super important for development. So that happens from one cell. So you can imagine if I'd got my Lego set back in the 1980s and opened the box and it had one brick and then it did this, it would have been amazing, right? If that set just emerged from one brick, it would have been amazing. Daphne emerged from one cell, and she is also amazing. But so, like, that's development, okay? Development is this process of differentiation. The cells, there's more and more cells, they specialize more and more, and they end up being this final product. So how's that possible? So really, the thing underpinning all of that is the genome, okay? So the genome, to continue my Lego analogy all the way through, that's the instructions. It's like all of the instructions for every Lego set that you could imagine, okay? All piled up. But somehow, from that, there's a heart, there's a brain, okay? So somehow the genome manages to coordinate itself so that these different things can emerge. And again, just to give a very simplistic view of what the genome looks like, but so you can understand like how this process takes place. So the genome is written in DNA, so that's bases of A, C, T, and G. It's about 3.2 billion bases long. That means that there are about 60,000 genes. 20,000 of them are what we call protein coding genes. And they're the ones I'm going to focus on today. That's not to say the others aren't super interesting. They're just the ones I'm going to focus on today. So the first thing that happens is that copies of those genes are made. They're transcribed into mRNA, and those mRNA are the instructions for how to make the proteins. They're the bricks. They're the things that, that our bodies are going to use to make a cell. And actually, all of our cells have the same genome. They all share the same genome. So the instructions for the same set of bricks is in every cell, but not all of our cells are the same. They're different. So how does that happen? So there are these actors. They're called transcription factors. They're also proteins, and they are the main coordinator of this choosing about what happens. Okay, so just to give you an idea of what that's like, here's that same genome. The green thing is a transcription factor, and you can simplistically think of it like the genes that have the transcription factor are turned on, so they get the mRNA, they make the bricks, and then the bricks will assemble into, in this case, a muscle cell. But a different transcription factor, different genes are turned on, different bricks, and what you end up with is a neuron. Okay, so these transcription factors are the things that coordinate which things are on and which things are off. And that coordinates which bricks are there and which bricks aren't there. And once the right bricks are there, the cells assemble themselves into these different cell types that make up our body. Okay, so just to remind you, I was explaining the development. That was the point of that. So we started off in this one cell, all of those bricks. By the end of a pregnancy, there's got this incredibly complicated system of cells. So just in the brain, this is some neurons that arrange themselves. Okay, so that was 2 minutes of an introduction to biology, and it is indeed a whole module. So sorry that I rushed that into 2 minutes. But the important things that I want you to take away from that is that development is this coordinated expansion of cells. It's an incredibly complicated process, and that transcription factors are an important regulator of what's going on while that's happening. So you can imagine that biologists thought of this as one of the most complex control problems that exist. Okay, like we go from one cell to a human, and it's all coordinated by transcription factors. And the reason that I'm saying it like that is because that is what's going to make you realize how profound what Shinya Yamanaka discovered was. So what Shinya Yamanaka discovered was this. So if you take one cell from a human, he took a fibroblast, which is a cell from your skin, and you just put 4 transcription factors into that cell that wouldn't normally be there. Okay, that cell will by itself turn back into an embryonic stem cell. So it completely changes its state. It stops being a skin cell. It starts being an embryonic stem cell. But this just completely undoes all of that complexity that I told you about. And it happens just by introducing 4 regulators. Okay, I showed you my multi-ant colony optimization controlling of networks before. I can tell you that it takes a lot more than 4 things to control the road networks. And here, this incredibly complicated process gets reverted by these 4 transcription factors. Also, this happens when Shinya Yamanaka did it, it took about 25 days. People have done this now in about 9 days. So these 4 things undo a 40-week process in 10 days, and it's only 4 regulators. So for me, that was an incredible discovery. The Nobel Prize Committee graciously agreed with me and gave Shinya Yamanaka the Nobel Prize for that. But part of the reason for that is that everyone was obviously very excited that if you could make embryonic stem cells, that gives you the opportunity to then make anything else. So I could take my skin, I could make embryonic stem cells, I could then differentiate them into any of these other cells and use those for treatment or use them for modeling disease. Okay. But there's something I should tell you about what happened. So this is the paper, the first paper. They did it in mouse to begin with. So as unfortunately is always seems to be the way. So Shinya Yamanaka, he won the Nobel Prize. Takahashi-san did all of the work. So here is Takahashi. This is my picture of him. So they had 4 transcription factors that did this, but they had no idea what they were. So what Takahashi did is that he tried combination after combination after combination after combination until he found the one that worked. Okay, and that took him about 6 years. Okay, and all credit to him for sticking at it and finding it. But just to say that, like, it's an amazing discovery, but the process of that discovery was rather long. Okay. But what was even more amazing is that it didn't stop there, actually. So I'll show you another experiment. So here is a microscope. If you look down that microscope, these are fibroblasts, same cells that Shinya Yamanaka used to make iPSCs. So then you take a different 4 transcription factors. So not the same ones that Shinya Yamanaka used, but a different 4. And you put them into those fibroblasts. And then you wait. You wait for about 2 or 3 weeks. And you put them back under the microscope. And then what you see is this. So this is exactly the same cells 2 or 3 weeks later, and you'll see that they're beating. So that's because those fibroblasts have been reprogrammed into cardiomyocytes. So again, fibroblasts, add some transcription factors, those cells become cardiomyocytes. They don't become embryonic stem cells, actually. They just convert directly from skin to cardiomyocytes just by introducing those 4 transcription factors. Okay, so what it looks like, that's what we really think now, is actually cell type is completely plastic, and you can convert between cell types. What you really need to know is what are the combinations. Oh, I should have put that up first. It's pretty awesome that you can convert between those. Because imagine, like, if you can convert between any human cell type, you can make cells that people have lost. You can convert disease cells back to a healthy state. You can make cells that people have never seen before. But there's one problem, which if you think about what Takahashi-san did, you'll start to realize. So there are thousands probably of unique cell types. Here are just some of them. Okay. And there are about 1,500 transcription factors, okay? And I'm not going to put any equations in, but let's just do a little bit of thinking. Let's assume that there is 1 transcription factor recipe for every cell type. That's not true. We know that's not true. Let's assume that each recipe uses 3 transcription factors. That's not true. Some of them require more. And let's assume that we can limit the search to 100 transcription factors for every cell type, and that there are 10,000 cell types. So if you just do some maths, that means we have to try a lot of combinations for every cell conversion. For all of the cell types, that's going to be an awful lot of trial and error experiments. And if we do it at Takahashi-san's rate, even if we get a team of 100 scientists, it's going to take 300 years till we find out what all of those combinations are. Okay, so we needed another way. Excuse me. We knew it wasn't going to be possible to do this by trial and error. Okay, so it won't surprise you, given what you heard about me, that the way that we tried to solve this was by using machine learning. Okay, but the first thing we needed was data. Okay, we needed to know if we had any hope of solving this, we needed to know what was the gene expression, which bricks were in all of the different cells in our body. If we didn't know that, it was going to be a very hard place to start. But luckily, Riken, this is Riken Yokohama, just south from Tokyo. The team in Ricken, they had developed a new technology called CAGE. For the molecular biologists in the room, this is a CAT. Now, it's a gene expression. It's like an RNA sequencing, but you only sequence the first part of the transcript. And they developed this technology, and they wanted to use it to work out what the gene expression, what bricks were in all different cells. So that was really great timing. So I was very lucky. I got this JSPS scholarship to go and work with this team led by a Swedish guy, Carsten. And we joined what was called this Phantom 5 consortium. So this was an international effort of both experimental biology people and computational biology people to try and catalogue gene expression across all cell types in human. Like today, that seems like you could do that in a single cell sequencing experiment in no time. At this time, this was a pretty ambitious effort. So here I am, a bit blurry, looking a bit grumpy. Here I am a few years later, still blurry, slightly less grumpy. Okay. So what did they do? So to put it very simply, to keep using my brick analogy, here was my genome with all of the bricks. What CAGE let you do was, in all of the different cell types, I could know how many of each of these different things was in each of these different cells, okay? And that's what the Phantom Consortium tried to do. Again, for the molecular biologists in the room, actually they were trying to find promoters and enhancers. But it also meant that we knew the gene expression of everything because of that. So that's my cartoon. Here's an example of what the data really looked like, okay? But this was really an important first piece of this puzzle. So now we knew what the gene expression was in every cell type. But like I told you, we have many thousand cell types. There's many hundred or many thousand different transcription factors, and it's impossible to test all of the combinations. So we decided to try and solve this by writing some computer program to predict which things we needed. This computer program is called Mogrify, and it uses the information that came from the Phantom Consortium, this CAGE data, and it also used some other information, like it used information about how things interact, so it knew which bricks were next to which bricks, and it also used some information from the genome. And then it took all of that, and for any possible conversion, it would make a prediction. I'm not going to talk too much about how it works today. We don't have enough time. I'm already way behind schedule. But these are the sort of three steps. The first thing it did is it worked out which things were specific for every cell. So that's this differential expression step. It then worked out for every transcription factor, it tried to work out how much influence it had on that cell type. Okay? And then it looked for sets of transcription factors which weren't redundant to each other. So they regulated different sets of things. The sort of idea behind that was that we wanted to find transcription factors that if we introduced them, they would very likely have a strong effect on the cell type that we were trying to make. So I sometimes put this here. This is to here square. And this was a revolution that was quite different to other revolutions because it was a revolution that was sort of a social media revolution. So the people that drove this revolution, they weren't like the most like polar, but they had an awful lot of influence. They were the ones that could use the social media to start this revolution. And that's exactly the kind of transcription factor that we knew that we wanted to find. Okay, so glossing over several years of work, we made this algorithm. We always had this idea that we wanted to be like a travel website. You could come in, you say where you want to start, where you want to end, and this algorithm would run, and it would make a prediction about the transcription factors that you needed to do that conversion. That's what we did. Okay, so we make this algorithm. It's supposed to predict how you can convert between any two cell types. So at this point, I should have said it's me and Julian. Okay, and we're super excited. We finished this tool. We write to the journals. Dear journals, look how great our tool is. They say, so what? It's just a computer program. We have no idea if it's worked. Sorry, I'm not interested. Okay, not more than one journals. I should. So which in hindsight, looking back, was fair enough. So what we did is I set out on a one-man campaign to go to as many stem cell meetings as I could and ask people if they do the experiment. And this is this particular meeting changed things a lot for me. So this was the 12th annual meeting of the International Stem Cell Society in Vancouver. I'm just going to point to the date to you for a second. I'm going to come back to that. I don't have any photos of this particular evening, which I'm very sad about. But I went to dinner in this teppanyaki restaurant up there. And those of you who've been to conferences, you know that you sort of awkwardly end up going for dinner with people you sort of half know, sit down next to people and tell them. So on this particular evening, I was super lucky because I sat down next to this guy. This is Jose Polo. And actually, I've been working with him and I talk to him almost every day since this lunchtime, this dinner. Okay, and let me explain why. So we had dinner. I explained to him that I'd written this algorithm, that no one cared because we hadn't validated it, and I was actually complaining to him that no one wanted to do the validation experiments. And he said, well, I'll do them. So here's another email exchange. I wrote to him the next day. I said, it was really nice to meet you. You mentioned last night, and he said you do some experiments. He just replied saying, sure, I'll get someone on it as soon as possible. Okay, so 3 months later, obviously, a lot of phone calls happened. He wrote to me and said, hey, it worked. Okay, so I'll show you the data that some of the data that he sent me, right? So we chose to try to do some cell conversions that no one had ever done before, okay? So we said, okay, let's try and take a keratinocyte and make a vascular endothelial cell. We put that into into the website I just showed you. It gives this prediction. And Jose did that experiment. You sort of just have to believe me, but I'll show you. I don't want to go into it in too much detail, but you can see here, like, so these things start off as keratinocytes, and this is after the experiment. So you can see the green ones are the ones that got the transcription factors, and you can see that they start to take this microvascular endothelial cell morphology. So that was the first one he tried. He also said, well, let's try and make keratinocytes. So he took fibroblasts, he took the set of factors that the tool predicted, he overexpressed them, and it worked. So again, where's my, here we go. I just point you to here. So keratinocytes, they have this very, very cobblestone-like morphology. Okay, so the green ones are the ones that have got the virus. Fibroblasts have a very long, thin morphology. And actually, you can see that in this particular place here, there's one cell that hasn't got the virus, and you can see that it still has the fibroblast morphology, but all of the things around it that did get those factors now look like keratinocytes. So they had changed. Okay, and like this was completely incredible, to be honest. Like I never thought that it would work quite this quickly and quite well. So it was published, but the main reason that I'm putting this up is to say this. So this was published in March 2016. So I met Jose in June 2014. Okay, so remember I told you that Takahashi took 6 years to do it. So we did 2. Actually, by the time the paper came out, we'd done about 6 cell conversions in just over 18 months. So this was really a demonstration to us that that this thing worked, that somehow the tool was able to make predictions that were reliable. I'm so far behind schedule. Okay, so then what? So like obviously we got a really nice paper, and as an academic, we always want to get nice papers. But I think that between the 3 of us, we knew that like we shouldn't stop there. So we tried to convince our university to start a company. They weren't interested. We tried to convince other people to start a company. They weren't interested. So we thought, well, we were going to have to start a company. So we did. And I'll tell you a little story, because I know sometimes in a presentation like this, it sounds like I always know what I'm talking about. But that's really not the case. It's the case at the end, but at the beginning, it's not the case, okay? So we wrote to loads of investors. Dear investors, look how great our tool is. So many investors. looks good, no thanks. So we keep asking, and eventually one says, looks good, let's do it. So we launched the company. Initially, it's called Cellmogrify. Actually, the only argument that me, Julian, and Jose have ever had is about what color that logo should be. And it was quite a heated argument. But that's not the story I wanted to tell you. So we had these investors. They said they wanted to invest in our company. So at the time, I lived in Singapore. Jose lived in Australia. These investors were from Wales. So we flew over back to the UK. We had the meeting with these investors the next morning. And they phoned us and said, oh, everything looks good for tomorrow. We just need to know the details of your lawyer. And we said, sure, no problem. We'll send that over to you straight away. We didn't have a lawyer. So we asked around for some friends, anyone we could find, if they could give us the name of a lawyer. We phoned a lot of people. Surprisingly enough, most people were busy the next morning. But eventually we got the name of someone. We phoned them up. They said, yeah, that's fine. I can do it. I'll see you there tomorrow. So we phoned back to the investors. They would say, oh, yeah, here's the name. And they're like, oh, we also need their address. And we're okay, we don't have that. We'll get it for you. We tried to phone the guy back. He didn't answer the phone. So we're like, okay, probably we can find his address just by searching him on the internet. So we searched him on the internet, and this is what we found. So the reason that he was free is that he had been struck off. But at this point, we had no choice. So we arrived the next morning with our lawyer, and we went into the meeting. We negotiated everything. Actually, he was really helpful. And in the end, we got the deal signed. But I just wanted to say that because we had no clue what we were doing. All that we knew was that we wanted to make this thing happen. And sometimes you don't know. Sometimes you just have to go with it. And that can sometimes work out. So this is the three of us actually on that morning. We're actually standing outside the guy's office, which was actually just his flat, before we went to the meeting. But that really was like the start of how we were going to turn this into something else. These are our first two employees. And actually, Joseph here, I think he must be the person in the world who has done the most cell conversions of anyone. Because basically what we used that money to do was to try to fill out our patent with as many conversions as we could. And he did all of these. And this is all in 18 months. So we did all of these cell conversions. Joseph did them all, actually. And bearing in mind that I told you that Takahashi did one in 6 years, Joseph did, I don't know how many this is, in about a year. And that still blows my mind today. But also just to say again, the speed that everything was happening was incredible. So now we're talking about June 2017. So between this and meeting Jose was still a short number of years. It was 2014 that I met him. The company has continued to grow. This is the office that they were at. Here's the team a couple of years ago. Here's us at some meetings talking about science. I did just want to show you one of the things that the company is doing. So one of the things that they're trying to do is to use this reprogramming technology to replace photoreceptors in the eye. So a very common cause of sight loss is that you lose the photoreceptors. But what we had was a technology to say, okay, well, let's convert something else in the eye into photoreceptors as a way to restore them. This is data from the company. So the first thing that I show you, so these things here are organoids of the retina, and they have these cells called malleable in, and also these, you can get rid of the rods. And the company takes the malleable, they overexpress, so that all these things here are malleable, they overexpress the factors and they start to convert into cones. They also now take mice, they do something to remove some of the rods from the the photoreceptors from the mice, and then convert Muller glia cells in those mice back to photoreceptors. And again, we're so far behind, you just have to take my word for it. Here are some cells that were Muller glia that have become photoreceptors. And what the company is doing at the moment is trying to demonstrate that by having done that conversion, that those mice recover their sight. Okay, so I'm just going to go over this super quickly, but the company's done a bunch of stuff. We've used the algorithm now to do loads of other things, like from the point of view of what's a cell, it could be anything. It can be a young cell and an old cell. You can convert between those. A cancer cell and a differentiated cell, you can convert between those. So please come and ask me, and I'll tell you all about the other things that we've done. Okay, I'm just going to go right on past that. Okay, so let me come back to this guy, Shinya Yamanaka. So what do we do next? So Shinya Yamanaka, like I just told you, he made this discovery, okay, that you can take a fibroblast and you can turn it into an embryonic stem cell. And people were doing this like over and over again. Actually, we had very little idea about what was actually happening. So we had very little idea about what was happening when you overexpress them, those factors. How did they become a thing? I put this up into some of my slides until I recently found out that actually these days not everyone knows what a phone box is. So, but it was a bit like this, like Clark Kent would go in, there'd be this, and Superman would come out, right? But we didn't know what was happening in the phone box. Okay, and that's what we decided to try and set out to do. Like, could we find out what was happening while the cells were undergoing reprogramming? And the technology that came along that allowed us to do that was this thing called single cell RNA sequencing. I'm going to use an analogy to tell you about why doing that, how that technology let us say something about this cell conversion. So here's the common. And what I've put on here is a start and a finish. Okay, so that's like saying here are the fibroblasts, here are the embryonic stem cells. Okay. Oh yeah, I made a diagram like that. There we go. If I put one runner on this track, you can see it tells you something about like where like the process from going from start to finish. Okay. But it still doesn't really tell me about like the routes that the runners take or like the decisions that they make during this process from start to finish. If I put 10,000 runners on that track, you actually start to get an awful lot of information. You can see like where the track is, you can see where some people get it wrong, you can see some cycles on there. Like this tells you quite a lot of information about the routes that the runners take, okay? And so we can do the same thing with single cell RNA sequencing. We can convert loads and loads of fibroblasts to embryonic stem cells, and then we can subject them to single cell RNA sequencing while that's happening. Okay, so we get to know about what their gene expression profile looks like at some point during this conversion. And then we can put all of that data on top of each other, and we'll get something that looks a little bit like this map of the common, like the routes that the runners take. Okay, here's my squiggles to say what I just said. So here are all the routes that runners take during this thing. So we want to know all of the routes that fibroblasts took during reprogramming. So we set up this experiment, and just to remind you that what we're trying to look at here is fibroblasts being converted to embryonic stem cells. So we thought we were looking at this conversion from here to here. Okay. So let me show you some data. I'm going to start going a bit faster. So here are imagine this like the picture of the common that I just showed you. Every dot is a cell, and what you can see is that I can see something about the paths. So the paths that you should look at to start off with, this blue and this orange path are the ones that we really expected to be there. They're the fibroblasts that are going from fibroblasts to naive or prime pluripotency. So these ones on the right-hand side are embryonic stem cells. Some of them also fail to reprogram. So there's this third path here, this refractory path. So they're fibroblasts that don't reprogram. So that it starts to look like this. But what I really want to draw your attention to is there's this one other path here. This path here. So these are cells also undergoing reprogramming, and they do something that we completely did not expect when we did this experiment. And let me just remind you about something. So we thought we were making the epiblast, these cells in the middle of the blastocyst. But actually, it looked like these cells, they looked a lot like the cells on the outside of the blastocyst. They looked a lot like the trophoblast cells. So actually what it looked like was that during reprogramming, some cells were becoming the cells of the epiblast, the middle, but also some cells were becoming cells of the outside, the trophoblast. Bearing in mind that at this point, hundreds of people had done this experiment, and we just hadn't noticed this until we did this fine-grained experiment. Okay, luckily for us, someone had created a culture media for trophoblast stem cells. So we took these cells that were undergoing reprogramming and we put them into a media that could sustain them. And they grew. They grew like they were trophoblast stem cells. And then we do lots of things, but I'm just going to show you this one experiment because for me, I had no idea that this is how you could do something like this. I don't know if you know, but if you take a pregnancy test, actually you're testing for the presence of a placenta, not for the presence of an embryo. So the postdoc went and bought loads and loads of pregnancy tests, and he took the media from those cells and he put that onto the pregnancy test so that he injected them into a mouse. He let them grow. He took the urine from the mouse and put it onto a pregnancy test, and that comes back positive because those mice have grown a human placenta basically inside. So they really are placenta cells. So completely not what we thought, well, not what we were expecting. And actually, what is incredible is that is not where the story finishes. It's even more unexpected than that. So that made us go back and look. Okay, so these are all. Imagine this is my top-down picture of the runners. These are all of the cells that are in in the reprogramming. So we knew there were some cells from the epiblast, this bit in the middle. We found out that there were some cells on the outside, and actually when we looked really carefully, there was actually also some from this third layer that emerges, was this primitive endoderm layer. So actually during reprogramming, all those fibroblasts, some of them became a different piece of the whole blastocyst. Okay, and we just hadn't noticed that. I can't find the original photo, so I only have this very stylized thing. But the very first experiment we did is we put the embryonic stem cells and the trophoblast stem cells together and cultured them, and they organized themselves. So you can see that there are the green ones are the embryonic stem cells, the pink ones are the trophoblast stem cells, and they're not universally spread. So then, as a result of that, we thought, well, I wonder what happens if we put the cells that are undergoing reprogramming into a 3D environment, something that could sustain a blastocyst. So we did, or Jose did. And what's completely incredible to me still to this day is that those fibroblasts, we induced reprogramming, we put 100 of them into this microwell plate, and they spontaneously form what looks a lot like a blastocyst. Okay, so these cells, they organize themselves into this incredibly complicated structure. Again, you have to take my word for it, but we characterize these things. They look a lot like blastocysts. We did all kinds of imaging to make sure that was the case, sequencing, all kinds of things. Okay, so what that really meant is that not only could you make embryonic stem cells, but you could take skin, fibroblast cells, you could put those reprogramming factors in, and what you'd get out was something that looked a lot like a blastocyst. Okay, so we had no idea that this is what we were going to find, but it's still incredible to me that that's the case. To go back to my analogy, to give you an idea, imagine that you went to the common to watch some runners. This is what you were expecting to see. This is what you saw, right? It's that level of surprise to us that we could get these things would self-organize themselves into something that we just did not imagine could be the case. Okay. I'm just trying to decide where to go. This was published, got a lot of press. Here's Jose needlessly standing in front of a screen full of pictures of blasts for the Guardian. One thing I did want to say is that actually this work is really important because it means that we can start to study human development in a way that hasn't been possible before. Okay, so it used to be that if you wanted to study human development, the only way you could get material would be by taking blastocysts that people who were doing IVF didn't want anymore. You could then use those, but now you could make them in a dish. But obviously, like that has a lot of ethical things to consider. Actually, in Australia, where we were doing most of these experiments, it was illegal to do human cloning. So actually, Jose initially was very worried that he was going to go to jail. But everything was done under complete ethical approval. And actually, we had some very unusual supporters. The Archbishop of Sydney came out in voicing his support for the work we were doing. And that's partly because these models meant that we could study development, and they were just skin, essentially. They were skin that was converted into something that looked a lot like a blastocyst. I'm way out of time, aren't I? What's the time? Is it? Can someone tell me the time? Okay. So, like I said, like trying to say about something that's not happening. I'm saying a lot of things that aren't happening. I wanted to talk about one thing that is happening. So, this is a project that I started in Duke NUS and that we've carried on here in Southampton. So, one of the things is that if you do single cell sequencing, not only do you get to know these parts, but you also get to know a lot about what the cells are doing. They don't all do everything at exactly the same rate. Same way that like some runners are faster than others, some like puddles, some do like puddles. If you watch this, if you can learn a lot about like the rules that they're following. And I'm going to go really quickly and just say we made this tool, Cell Tuner, and one of the things that we've been working on is using that to do something like this. If you've got a cell state that you don't want, how can I push that to be a cell state that you do want? So I've been Disha working with Sian. Those guys are sat over there. We've been trying to use that to say, well, some patients do respond to treatment and some patients don't respond to treatment. So can we use these ideas to try and convert cells from patients that don't respond to a treatment to look more like the ones that do respond to a treatment? So I don't take loads of time. Please, I invite you to come and ask me or those guys about that afterwards. But it's really looking like it's starting to work. We can take these T cells from patients that don't respond and convert them into cells that do respond. Okay, Phil already mentioned this. Like one of the things that we're also starting now is we have this partnership with the Rosalind Franklin Institute. I just very, very quickly tell you about why I think that's so exciting. So I put this picture up earlier on, which was like how complex a cell is, right? How many bricks are there? And actually, if you do omics, pretty much all you get to know about is the number of bricks, okay? But actually, a cell looks like this. It's structured, and an organoid looks like this, right? It's just all cells coordinating themselves. So what the Rosalind Franklin Institute is very well known for are imaging technologies that let us look about not only what bricks there are, but where they are in relationship to each other. So the idea is that we use technologies they have, like this is cryo-electron tomography, and you can see individual proteins here, where they are, what they're next to, what organelles they're forming. So we're going to try and use this information to build not only models about like abundances of bricks, but also where they are all positioned in time. All of these people here are doing awesome things. I wish I had more time to tell you all about it. Some of them are here. You should talk to them about that. I'm just going to go super fast. One other thing that I'm really happy and proud to have also started. So here's my dad. This is my mum. She very sadly was diagnosed and died very quickly with pancreatic cancer. So I'm a researcher. So I thought I should want to do something about this. So with all of these founders, all of these funders, and a whole bunch of other people across the UK, we've recently launched this thing. It's called Cancer Data-Driven detection. So that's using some of these ideas that I've been telling you about, but actually, rather than trying to predict what to do, try to predict what could occur that means that you would end up to have cancer. Yeah, so I thought I would finish with what does the future look like. I'm now a professor of AI for the life sciences, so I should say something profound, I guess, right? But like for me, it's amazing how quickly this world is changing, right? So ChatGPT is 3.5 years old, and it's already everywhere. AlphaFold is 5 years old, and we just came back from a conference, some of us yesterday, and people are now using AlphaFold-like technologies to design proteins that very specifically bind to individual proteins of interest. So they can design drugs using these programs. And they really, really are doing that. I'm nearly finished. I promise I'll shut up soon. Like I showed you Mogrify, like the complexity of those models is now getting much, much higher. So we can now really predict how cells behave, and I think that's going to change very quickly. I thought I would say some advice that I would give to myself a while ago, or to young scientists now. So based on my experience, at least, these are the 3 things I think you should really think about. The first thing is that it's really important that you stay curious and you keep trying new things. The second thing is technology. One of the things about technology moving so fast is that it could be really easy to not be an expert anymore. Right? You can ask your computer anything and it will tell you the answer. But you shouldn't let that be a reason to not become an expert in something. And the last thing is that you should enjoy it. And I really have. I just very quickly just show you some pictures of like things that have happened, things that we've done over the years. And I've really enjoyed every bit of it. I wanted to say also one thing though, like this presentation is really a description of all the things that have gone right. Sometimes things don't go right. And then you also get to see how good people can be to you. This is Max, Lindy, and Phil. At various times, they have stood up and helped me when things haven't been going well. And I'm super thankful for that. Obviously, I'm also really thankful to my family who are dotted around the way. There's you. They always support me to do this. Alice thinks it's a joke that whenever we go on an aeroplane, a journal emails me saying I've got 2 days to do the proofs, but she always lets me do the proofs. So thanks very much. I'll go right back to the very beginning. Here's me with some bricks, and here's Daphne with some bricks. So maybe in a few years, she'll be standing up here and I'll be standing over there telling her to shut up. So thanks very much. Thanks for listening.
Thank you very much, Owen. Do we have time for a couple of questions? Yeah. Okay. Questions?
Oh, well, over here.
I was going to ask. You say it. You can say it. Oh, yeah. No, you know how to do it if you want to. I don't know. We've got another one in the meantime. Go on then.
I think we wrote our names or something really unimaginative, like Owen and June were here or something.
Any questions? Yeah,
so on the run, when I take a run, I'm a very bad runner. So from the start, my perception of how this run is going to go and which way I'm going to go is probably very negative. So you talked about how you're going to be able to adjust people's journeys, those runners, those cells, and negotiate them through that. Where do you see that going? Will you convince the bad running cell like me to go the right way, and will you tell them before they go, or during their journey, or afterwards? Does that make sense?
Yeah. Well, so hopefully any of those things is possible, right? Like the perfect thing is that you would intervene before something happened, so that it couldn't happen in the first place. But I think we can also take things that have happened and undo them to some extent. I think that will be possible. Like it's super plastic in space.
Thanks, Owen. That was a wonderful talk. My question is, when machine learning predicts something that doesn't validate in the lab, what do you do and what does it tell you about the data and the process and so forth?
Yeah, this is a very, very, very important thing actually, because one of the things that we've been guilty of in science is that we always publish our positive results. And actually, from a machine learning point of view, the negative results are just as powerful as the positive results. So I think that what you'll start to see is that like more and more of that gets published. Because actually, if you know that it didn't work, you can retrain the model so that it can correct itself. Like in some senses, like it's much easier to change how models work than have like our own understanding of things. So,
If not, I have one. I would be curious to know your intuition where the next, like what directions would the next breakthroughs come from. Do you think that's mainly just scaling up AI data centers or the technologies, or maybe experimental methods that give us more resolution, higher resolution into biology?
So I mean, like, I think that all of those things are important. Like, I think that we will, like, data centers are going to get bigger, algorithms are going to get better. But actually, like, they're sort of the means to an end, right? Actually, for me, like, the next big breakthrough will be that at the moment, I think now that, like, we are today, like I said, we just come from this meeting, I think that we can, like, computationally engineer proteins now. I think that in the next couple of years, like, you'll start to see more and more examples of, like, proteins made by a computer that do things. And I think my work and the work of other people says that that's also probably now getting close to individual cells. The next big thing will be like, okay, well, can we build artificial biological systems of cells that do things that are super beneficial, like manufacture drugs or those sorts of things. Like our body, I'm not one cell type, right? Like I'm a system of cell types. And I think we will be able to start engineering systems of cell types that can do things.
Doing what you love, building things.
Yeah.
Okay, I think that's my question. And it's that she knows somebody with the Lego book that you all have been built together. Is that right?
Thanks, darling. We can do that. Yeah.
Okay, if that's the last question then. In that case then, I think you join me with thanking you again for a wonderful talk.